Understanding the complexities of Cornelia de Lange syndrome, CdLS, in the Chinese population is a crucial step towards better diagnosis and management. Imagine a world where a rare genetic disorder, characterized by distinct facial features and developmental delays, affects individuals differently based on their genetic background. This is the reality for those living with Cornelia de Lange syndrome, and recent research aims to shed light on how it manifests in Chinese children. Cornelia de Lange syndrome is a rare genetic condition marked by a range of symptoms, including distinctive facial traits such as fused eyebrows, a short nose with an upturned tip, and downturned mouth corners. Other common signs include growth retardation, a small head size, microcephaly, developmental delays, cognitive impairments, excessive hair growth, and limb abnormalities. To date, scientists have identified mutations in 7 specific genes: NIBPL, SMC1A, SMC3, RAD21, BRD4, HDAC8, and ANCARD11 as being linked to CdLS. However, the specific clinical and genetic characteristics of CdLS in the Chinese population have remained largely unexplored until now. A groundbreaking study led by Professor Chun-Siu Gong from the National Center for Health in Beijing, China, aims to fill this knowledge gap. The research, published in Pediatric Investigation on July 2, 2025, provides valuable insights into how CdLS presents in Chinese pediatric patients. Professor Gong emphasizes the importance of this study, stating, "Estimating the prevalence of CdLS has been challenging due to underdiagnosis or the failure to recognize milder forms of the syndrome. By understanding the clinical and genetic aspects of CdLS in the Chinese population, we can equip pediatricians with the tools they need to improve diagnosis and care." The researchers conducted a comprehensive analysis of 19 Chinese children diagnosed with CdLS. They meticulously recorded each patient's clinical symptoms and disease severity before extracting DNA from blood samples for whole exome sequencing. This advanced genetic testing technique allows scientists to analyze the protein coding regions of the genome, providing a detailed view of genetic variations. Based on a standardized scoring system, 12 of the 19 patients exhibited classic CdLS symptoms. 16 children showed global developmental delays, while 14 had prenatal growth retardation and short stature. The most prevalent clinical features included craniofacial anomalies, particularly a short nose and upturned nose tip, along with small hands and feet. Less common symptoms were skin irregularities and hearing loss. The whole exome sequencing revealed 19 mutations across 3 genes. The majority of these mutations, 15 in total, were found in the NIPBL gene, which codes for a protein called delingin, crucial for development. These mutations included various types: missense mutations, which change a single amino acid, nonsense mutations, which cause premature termination of the protein, frame shift mutations, which alter the protein sequence, and splicing variants, which affect mRNA processing. Additionally, 2 new variants, 1 missense mutation and 1 splicing variant, were discovered in the SMC1A gene, which regulates chromosome structure. Similarly, 2 new variants, a frameshift mutation and a deletion, were identified in the RAD21 gene, involved in cell division. The study also explored the relationship between genetic changes and clinical manifestations. Notably, patients with NIPBL null variants, which result in the complete loss of gene function, exhibited significant growth impairment and microcephaly. This finding underscores the critical role of NIPBL in the development and severity of CdLS. To investigate potential therapeutic approaches, the study focused on 3 patients whose parents requested recombinant human growth hormone, RHGH therapy. 1 female patient, diagnosed with CdLS at age 3, began RHGH therapy at age 5. Her height increased by 8 centimeters, but the treatment was stopped as her parents felt the response was insufficient. A male patient, diagnosed at age 10, continues to receive RHGH therapy and has grown 12 centimeters taller. Another female patient who started RHGH therapy at age 7 saw her height increase by 10 centimeters within a year, but experienced hand and foot enlargement, leading to the discontinuation of treatment. Lead author Xiaoqiao Li commented, "While RHGH therapy proved effective in 2 cases, it also resulted in undesirable side effects in 1 patient." This highlights the need for personalized treatment plans and careful monitoring of dosages to optimize outcomes and minimize adverse effects. By uncovering the unique characteristics of CdLS in the Chinese population, this study offers valuable guidance for clinicians in diagnosing and managing the condition. It also lays the groundwork for future epidemiological studies, potentially leading to more tailored and effective treatments for Chinese patients. What do you think about the implications of these findings? How might personalized medicine play a role in treating genetic disorders like CdLS? Share your thoughts in the comments.